in cancer research. Thank you so much for your time and for joining us here this afternoon. This special 2-hour symposium will host over 16 speakers representing about 10 institutions across Canada, and it will showcase the groundbreaking research addressing the complex challenges in cancer research from basic science to clinical translation.
Really, CHCCR celebrates the cancer research done by scientists, clinicians, and trainees across Canada. So really, this involves a series of short and engaging presentations with the challenge to effectively communicate complex research topics, creative activity, and also the broader impact to the Canadian cancer community in 3 minutes or less. So we'll start off the event with a keynote from Dr. Fred Dick from Western University about the hallmarks of cancer. Then we'll have 4 sessions. The first 2 will feature talks about basic science research, followed by a short break, and the last 2 will focus on clinical and translational research. Each session will have 4 consecutive talks, followed by an open question period. And we encourage you to ask questions using the chat function throughout the talks. Our Riot team moderators will read your questions and then direct them to the group or the appropriate speaker.
Finally, to make this event more engaging and to introduce some friendly competition, we're going to be asking you to vote for your favorite talks. So please keep that in mind as you're listening today. We thank you all for joining us here on Zoom. And just a couple of housekeeping items. We want to remind you to please keep yourself on mute throughout the duration of the event. And then as well, you are welcome to share your video, but we will be recording the talks. So you may appear in the video recording if you do choose to have your video on. Now, I'd like to pass it over to Camilla Zakirova, who's a member of the London Riot team, to introduce our keynote speaker.
Good afternoon, everyone. My name is Camilla Zakirova. I'm a PhD candidate at Western University and one of the moderators for this event. I'm very excited to introduce our today's keynote speaker, a remarkable contributor to the field of cancer biology, Dr. Fred Dick. Dr. Dick is a professor of pathology and laboratory medicine here at Western. His research focuses on understanding the molecular mechanisms of proliferative control and identifying how their misregulation is linked to tumor growth. His lab is particularly interested in the retinoblastoma tumor suppressor protein and its role in cancer progression and treatment response. Dr. Dick earned his PhD in biochemistry at Dartmouth Medical School. He then completed his postdoctoral fellowship at the Massachusetts General Hospital Cancer Center. Dr. Dick joined Western University in 2003. He holds scientific appointments with the Children's Health Research Institute and Lawson Health Research Institute. He is recognized as a distinguished scientist with the London Regional Cancer Program and is also a wonderful mentor to undergraduate and graduate students. Dr. Dick's extensive knowledge in cancer biology field is why we invited him today. We believe that his talk on the hallmarks of cancer will provide a great introduction to this event and help you appreciate the complexity of cancer. So without further ado, please welcome Dr. Fred Dick.
Thanks. I believe I'm unmuted now. Thank you very much. Thanks for the kind invitation to the Riot team, and thanks, Camilla, for the really generous and kind introduction. I'm going to tell you guys a little bit about the basics of cancer as we know it, really at a level that we often teach to undergraduates. That's been probably best encapsulated by the hallmarks of cancer. And so let me advance this here. Okay, so it was appreciated a long time ago that there was something special about cancer cells in comparison with normal cells. And I have basically 2 simple examples of this to show you here. So the first one is a basic histology image that looks like a normal tissue And individual cells have distinct shapes and relationships to one another that's important for tissue integrity and function. And it was appreciated very early on that cancer cells are a derivative of these normal cells that take on new properties that really don't follow these classical rules. So the shapes and structure and relationships with one another are lost. And so while these cells might have defined purposes and have some amount of discipline and organization within tissue, cancer is characterized by this unwanted cell growth and loss of integrity of tissue structure. So this is sort of one of the basic observations, but an important experimental insight into cancer came from early experiments done actually in some cases even 100 years ago, if you can believe it, where people took examples, they cultured and worked with normal cells or isolated samples from patients and cultured cancer cells and injected these into recipient mice. And the observation was that normal cells implanted into a mouse didn't form a tumor, and that cancer cells, of course, would form a new tumor when injected under the skin of these mice. And so this might seem a little bit obvious that normal cells don't form tumors and cancer cells do. But what it actually showed people is that cancer cells contain everything that they need to self-instruct the formation of yet another tumor. And so it gave rise to, and these experiments and a host of others gave rise to this basic idea that there must be simple principles that distinguish a normal cell and its normal biology from the type of cells that you find in cancer. And so this was the idea that was the genesis for the original concept of the hallmarks of cancer. And so Douglas Hanahan and Robert Weinberg proposed in 2000 by writing a brilliant review article that they entitled The Hallmarks of Cancer. They basically proposed at that time 6, what they said were sort of unifying features that cancer cells have that allowed you to distinguish a normal cell from a cancer cell. And these were essentially features that a cancer cell would have to acquire. So in order to kind of keep to time and fill this in about 10 minutes and be informative, I'm not going to tell you about every single one of them in a really didactic way, but I'll give you a few examples. I mean, cancers are characterized by unwanted cell growth. And so it was observed fairly early on that cancer cells have an ability to provide themselves with a growth signal so that they can replicate and proliferate all on their own without needing growth factor stimulation the way a normal cell would. And I think perhaps also important to relate it back to the mouse example that I showed you on the last slide, that cancer cells know how to instruct the tissues around them that in a way that allows them to recruit new blood vessels. So as a tumor grows, it needs to acquire nutrients and oxygen, and it does this through angiogenesis. And so the mouse example shows you that injecting pure cancer cells instructs the tissues around it to start allowing it to grow blood vessels and to allow it to form a tumor. And so these were some of the basic ideas that allowed cancer cells essentially to function. And so these ideas fueled the field for a good 10 years. And then along came a new generation of hallmarks. So 10 years later, the first 6 principles that we had for cancer cells that we called hallmarks seemed insufficient. And there were plenty of other examples of things that had developed out of that. So in 2011, a new Hallmarks Next Generation report was published, and it expanded from 6 features to 10 features, some of which became a little bit more encompassing than merely a cell autonomous function, although it does contain a lot of those. Items that I want to basically highlight here, I want to tell you a little bit about cellular metabolism. I believe there are a number of talks coming up this afternoon that are going to tell us about features of cell metabolism. So this is basically one of the features that's really important for a cancer cell, that normally cells in our body grow through a process called oxidative phosphorylation, where they break down sugars in a very complex way that generates the maximum amount of energy. Cancer cells, by contrast, tend not to do this. They tend to gobble up sugar in a very rapid and wasteful way. And this has a couple of important features. One is it allows the cells to grow quickly. But from a cancer perspective, it actually allows a lot of possibilities for imaging tumors. And I think that we're going to hear a lot about that in this afternoon's talks. It's the ability to distinguish a cancer cell or a tumor from normal tissue based on its metabolism has actually turned out to be really important in the world of imaging. Okay, and so then the last dimension or new dimensions of Hallmarks of Cancer that just came out in this past winter added 4 new hallmarks. And so again, without exhaustively talking about every single one of them, I want to highlight one of them, which is called phenotypic plasticity. And so basically, with all the rules we've come up with to try to explain why cancer cells behave the way they do, it turns out that they have this really wonderful feature called phenotypic plasticity, which essentially allows them to change the rules when they need to. So there are circumstances where cancer cells will encounter new environments, whether it's metastasis, cells travel to a new environment. They need to take on new properties in order to acquire new nutrients and to survive and proliferate in time in that location. And so phenotypic plasticity is actually really an important feature that, that my lab is particularly interested in. So I'll give you just one little snapshot of work that goes on in my lab. In fact, Camilla is an important member of the team that works on this project. Is that what's been observed is that in ovarian cancer, there's a very specific feature in which cancer cells paradoxically stop proliferating and enter a dormant state. It allows them to survive and spread. It allows them to resist the effects of chemotherapies. And this is key to how they function and how this disease progresses. And so this is a good example of phenotypic plasticity, that cells are sloughed from the primary tumor. They float in the abdominal cavity. They can organize themselves into these clusters. We call them spheroids, where they become dormant. And these dormant cells will essentially float around until they encounter a new microenvironment, usually a peritoneal surface somewhere else in the abdominal cavity. They can now colonize that and they can reverse their dormant metabolism and start to proliferate again. And so we've created and utilized mouse tissue culture models of this, where adherent cells mimic the primary tumor, cells grown in a very specific suspension type approach allows us to mimic the intervening states. And then we can also study the ability, or as a proxy of metastasis, their ability to sort of reattach. And this is some of the mechanisms that we're doing that build on the hallmarks of cancer and allow us to investigate some of the latest ideas in cancer. And so that's, I believe, the end of where I'm going to speak, and I'm happy to take questions if there are a couple minutes, or we can move on as our.
Thank you, Fred, for this enlightening and entertaining presentation. So I will read the questions from the chat from the audience, if there are any questions. Put your questions in the chat. So the first question is, do all cancer cells, i.e. for all cancer types, show all the hallmarks?
That's a really good question. I think that we should always remember that cancer is 1,000 different diseases. And so there's always an exception to these rules. And it very much varies based on cancer types. Probably the easiest way to illustrate this is that a lot of blood-borne cancers, based on the biology of the disease, don't necessarily have to have all of these hallmarks, which are probably more likely to be needed in solid tumor examples. So there are some easy examples of how there are exceptions to the rule.
Thank you. The next question is, what do future emerging hallmarks do you foresee in the future?
That's a really good question. I think really it depends on where research takes us. I sometimes think that an example of the phenotypic plasticity kind of speaks to the idea that maybe if we're coming up with hallmarks which essentially accentuate the fact that there can be ways that cancer cells can make themselves an exception to all the rules, kind of is the final feature that you need to, anything can be defined as being within phenotypic plasticity because it can fit whatever paradigm you want it to fit. So I wonder how many more years there will be of hallmarks. I mean, I guess time will tell if there's another paper in 10 more years that adds on a few more features, but I think that we're running out of ideas, but then again, maybe you could have said that 15 years ago. So time will tell.
Another very interesting question from Suzanne. She's asking, if cancer gobbles up sugar, should cancer patients maintain a low sugar diet?
That's a really good question. And so part of the problem with cancer cell metabolism is that they reorganize their metabolism in a way that allows them to become sort of the most important nutrient utilizing organ in the body, so to speak. So having low sugar means that the rest of your body will get less sugar before your tumor will get less sugar. So it's the observation that cancer cells tend to like to use, a fermentative, sorry, not fermentative, but a glycolytic pathway to utilize sugar. They do it in a way that robs the rest of your body. So that's probably not the way that we can really help ourselves. But it creates a vulnerability that is certainly in clinical trials to be attempted to utilize as a treatment approach.
Another very interesting question is, could cancer have once been an adaptive response to injury or toxicity?
That's a great question. So there are cancers. It's a great way to answer this. There are popular television programs, Dr. Pimple Popper, that talk about sarcomas. A lot of these benign sarcomas that are problematic for the people who have them often have their origins in some sort of acute trauma, that there was some sort of damaging event that led to kind of an aberrant repair event that led to these benign tumors. So there's certainly some evidence of how that can happen, but I'm not sure that you can really relate that kind of scenario to every cancer type. I think some of the more deadly types, it's harder to reconcile that as being the origin, but there certainly are cancers where that looks like that is the explanation.
Interesting. Another question, how do some cancer cells remain dormant for a long time, for example, 10 or more years, and sometimes not so long before metastasizing?
Okay, well, if we knew the answer to that question, we would really be taking a chunk out of cancer. And that's the sort of stuff that my lab is interested in. I mean, on the one hand, I'm very interested in doing ovarian cancer research and helping these patients. But I also think it's a good model of the longer term dormancy paradigms like breast cancer, where it can lay dormant for decades. I think it's a harder question to ask if something lays dormant for decades, easier if it's dormant for weeks, months, maybe, kind of thing. But I think if we could understand that, we could really improve treatment outcomes and survival in the long term. But that's the $1 million question, really.
For the interest of time, I will just ask one last question from Joan Romero. What are your thoughts on atavistic oncology, the theory that cancer unlocks a set of genes that were beneficial when our progenitors were single-celled organisms?
That's okay. That's a really great philosophical question. So how much time do I have, Camilla?
3 minutes. 3 minutes.
Okay. Well, I'm probably not even going to fill 3 minutes. So this is a question when I teach undergrad classes, I often discuss this, did evolution select for cancer protecting mechanisms, or, when evolution was really shaping the human race, did we not live long enough for that to be the case? And so I would say my personal feelings are that a lot of the things that I think are important in cancer, a lot of the sort of anti-cancer mechanisms like DNA damage repair, for example, I think a lot of these are shaped by evolutionary selection for survival of the species. And that what we see in cancer are these kinds of benefits and drawbacks of a mechanism that was really meant to serve our species over the long term, and what would allow us to survive and give rise to robust progeny that would also give rise to robust progeny. And that in some cases it's helpful in protecting us from cancer. In some cases it seems like it doesn't possess the things we need. But that's sort of been my personal philosophy on this, but that's just an opinion. I mean, we'll never really know the answer to it. But I love having these discussions about what creates our susceptibility and what are our best abilities to fight it. So thanks for that question.
Certainly, it's very interesting question. Well, thank you so much, Fred, for taking your time and giving this amazing speech today. Really appreciate it.
Great, thank you, Dr. Fred. It's interesting. I mean, it seems that every few years or so, we get a new update on the paper, the Hallmarks of Cancer. So in 10 or 15 years, when the new paper comes out, Dr. Dick, be sure to check your email for another invite. So hi, everyone. I'm John Miguel Romero. I'm an MD PhD candidate at McGill University working on the immune system in pancreas cancer. I believe my co-chair, Sandrine, is also online. We're the co-chairs of the Montreal team here in Quebec, and we have the absolute pleasure of presenting our first session, Basic Science, and we have 4 stellar talks headed up. So without further ado, I'll introduce our first speaker. It's Dr. Jessica Rogers, a current doctoral fellow, postdoctoral fellow at the School of Computing at Queen's University in Kingston, Ontario. And her talk is entitled Surgical GPS, Finding Breast Cancer for the First Time. Dr. Rogers, if you're on, you can go right ahead.
Great, thanks for the introduction. Up to 40% of women in Canada who go through breast-conserving surgery to remove breast tumors leave their surgery with cancer cells remaining in their bodies. These cells can spread throughout their body and regrow into new aggressive tumors. In the current method, this remaining cancer is only detected once the tissue has been sent for careful assessment after the surgery is complete. And so these women will be called back for additional surgeries. This disrupts the lives of these women and their families once again, reigniting cancer fears. And so this need for repeat surgeries puts heavy burdens on both patients and surgeons. What makes this type of surgery so difficult is that breast tumors are often non-palpable, meaning that they can't be felt. And so our surgeons can't see or feel the difference between the cancerous and healthy tissue. And so we've left our surgeons driving blind, relying on their experience and notes from images taken before they started operating. But what if we could turn on the headlights and let them see where they are driving, or in this case, what they're cutting through? Our research focuses on the development of an integrated surgical tracking system that allows our surgeons to visualize where they're cutting in real time relative to the tumor outline from ultrasound images taken in the operating room. We do this by attaching small sensors to the surgical tools and ultrasound probe, using an electromagnetic tracker to display their positions in space. And we've already brought this research technology into the clinic to be used in patient trials and have been able to show that the system helps our surgeons see where they're cutting and actually allows them to be more precise, taking out less healthy breast tissue. But we know that tumors are complicated and can have complex shapes that aren't always easy to see. And so we also want to equip our system with more tools to let our surgeons evaluate where they are cutting. To perform these procedures, surgeons use a tool called a cautery, which burns tissue as it cuts through it, sealing off bleeding. This creates smoke that we're able to suck up and perform rapid chemical analyses on. We've been able to use this to create chemical profiles of different tissue types and are investigating different ways of grouping these profiles, including using sophisticated artificial intelligence techniques. Using these signatures in the operating room, we can tell the surgeon what type of tissue they are cutting through and give them an alert if cancer cells are detected. This is our rearview mirror. It lets our surgeon check what they just cut through and enables them to go back and change where they're cutting to ensure that they are removing the entire tumor. Putting our tracking information together with this chemical information allows the system to act like a surgical GPS, giving our surgeons the knowledge to navigate to the location where cancer cells are detected and target suspicious regions. This instant feedback, while still in the operating room, gives the surgeons the confidence that no cancer was left behind, providing greater reassurance for patients and their families. This feedback also lets our surgeons take less healthy tissue, improving cosmetic outcomes for our women. Ultimately, we hope with this innovative surgical GPS technology, thousands of women can leave breast-conserving surgeries with the knowledge and peace of mind that all the cancer was removed the first and only time. Thank you.
Thanks, Dr. Rogers. That was a very concise and surgically precise talk. Wonderful. Up next, I have the pleasure of introducing Pradharni. He's a master's candidate at McMaster University in Hamilton, Ontario, Canada. And his talk is entitled Friends or Foes? The Toxic Relationship Between Fat Cells and Acute Myeloid Leukemia. Go ahead.
Thank you so much for the introduction. Acute myeloid leukemia is a cancer of the blood and of bone marrow and takes the lives of 1,100 Canadians annually. Older patients make up a majority of AML cases, yet have very low survival rates, and this is because of the lack of tolerable treatments. Fortunately, a new chemotherapy combining venetoclax and azacitidine, which I'll refer to as Ven-Aza, has shown great promise for these older patients, but chemo resistance continues to be an issue. Many studies have looked into how chemo resistance works and have pointed to the bone marrow microenvironment as a potential cause. The BM microenvironment consists of cells that surround the cancer, and of particular interest to our study are the bone marrow mesenchymal stromal cells, or BMSCs, and the fat cells that they give rise to, also known as adipocytes, both of which make up a majority of the bone marrow. Studies have actually shown that AML cells can manipulate these two cell types to contribute to their own growth and induce chemo resistance. However, the interplay between the microenvironment and Ven-Aza chemo resistant has yet to be studied, which is the objective of my research. The image on the left showcases my experimental design. And to sum up quickly, we grew AML cancer cells onto both BMSCs and an adipocytes. And we looked at how these co-cultures affected the cancer cells. And we looked at three things: proliferation, or the growth, the rate of growth, viability, or the percentage of live cells, and energy metabolism. The control for these experiments were just AML cells grown alone, known as the monoculture. The legend is in the bottom right under the mitochondrial metabolism graph. We did study this co-culture effects both in the absence of Ven-Aza and in the presence, shown on the top and the bottom respectively. So we'll start with the top. In the absence of treatment, AML cells don't like growing on adipocytes as they have lower levels of growth, proliferation, and viability, but these cells are somehow able to improve energy metabolism. Contrastingly, if we look at the bottom panel, these same cells, when exposed to the co-treatment, survive better when grown on adipocytes. So our data provides evidence for a dual role of adipocytes, where in the absence of treatment, it inhibits the cancer, but in the presence, it starts protecting the cancer. And for future studies, it's very important to understand what factors are at play here in this protective role of adipocytes so that we can target it and to potentially overcome chemo resistance. Thank you so much for listening. And if you have any questions, please don't hesitate to reach out.
Thank you, Pradharni. That's a very promising project. Excited to see results. Up next is a real treat. We have Dr. Sarah Machid. She's a current scientist in the Department of Bioengineering and the Canada Research Chair in Nanobiosensing Devices at McGill University in Montreal, Quebec, Canada. If Olha is on, or whomever is controlling, I will ask that the record Claire, if you're. Thank you, Claire. And last but certainly not least, we have Dr. Simon Rondeau-Gagné. He's an associate professor in the Department of Chemistry and Biochemistry at the University of Windsor in Windsor, Ontario. The title of his talk is Polymer Nanoparticles, Developing a New Molecular Target for Glioblastoma. Dr. Gagné, you have the floor.
Thank you very much, and thank you for the organizer for having me this afternoon. So for the next 3 minutes, I'm going to present you a perspective on cancer research from a materials chemistry perspective. So hopefully you're going to appreciate a different dimension to this research. So in my team, we're materials chemists, and our challenge, or one of our challenge, is glioblastoma. So I want to thank the speaker that went right before me to introduce the problem. I'm just going to give you my perspective, but as you found out already, it is an extremely aggressive type of brain cancer. The growth is fairly fast. It is highly heterogeneous and is really difficult to treat. So it's it's an outstanding challenge, and it is something definitely that needs a lot of research to be able to minimize and hopefully prevent. But there's also some drawbacks that I want to highlight on the treatments. So first of all, right now, mostly surgeries are the ones that can help to minimize, but relapsing is fairly common, unfortunately. There are side effects to some treatments as well. The drugs that we developed are really difficult to get to GBM because they either lack of specificity or they can't cross what we call the blood-brain barrier, which is kind of a natural barrier for the body to protect brain. So it is really difficult from a chemistry perspective to be able to design a drug that will either be specific to GBM or at least be able to cross that blood-brain barrier and reach our goal to get in the brain. So our solution as materials chemists is to use nanotechnology. So there are tremendous progress on drug design and drug discovery, but we think that nanotechnology can bring another dimension to this. And our focus is on polymers. In my team, we use the polymers that you see in the middle panel. It's called Dpp Based Polymers. And for those of you who owns a Ferrari, it is known to be the red pigment in the Ferrari color. So this type of materials are wonderful, outstanding for electronics, but you can also make nanoparticles of them. As you see on the middle panel, we can make nanoparticles of fairly small diameters, and we can wrap them up with a surfactant that help us to control their chemistry. So in my team, we use hyaluronic acid as a surfactant, and we decorate these nanoparticles with different types of functional groups to control their permeability through the blood-brain barrier and also their selectivity to GBM. So we're not the first one, obviously, to use these as treatments, but mostly they're used in the literature for photothermal therapy. So they use our little nano antennas to help to treat cancer, but we want to make them by itself a treatment for GBM. So on the right panel, just to go quickly, we did a bunch of different tests with our collaborators, look at their selectivity for GBM. We also looked at their use as additional treatment in different combinatorial therapies. And so far, we found that they are actually a really amazing platform that are selective, that can be treating or reducing the tumor burden for GBM, and they have a lot of promises for hopefully developing new platforms in combination with existing drugs to treat this cancer. So thank you again for the attention. I'm going to stop here, but I'll be happy to answer any questions. Thank you all.
Thank you, Dr. Rindeau. And on behalf of everyone, we'd like to take the time now to just clap, even if it is virtual, for all the speakers. That was an excellent talk. Excellent talks. Right now, I'll pass it over to Sandrine, my coacher, and she will moderate the questions.
If anyone has any questions, you're welcome to write them in the chat and I can read them out loud. We actually have one already. It's for our first speaker, Dr. Pack. It's for Dr. Rogers, actually. And the question is, have you looked into how this works for lobular breast cancer versus ductal breast cancer?
Yeah, so this is a fantastic question, and it's not something that we have explicitly looked at. What I can tell you is that our current data set is almost entirely ductal cancers, but we do have a few lobular cancers in there as well. So right now, mostly what we've done when developing our chemical profiles is basically we just look at normal or not normal, where normal is basically fatty breast tissue. And so anything that shows up as not normal fatty breast tissue is flagged as potential cancer. So my feeling is that it probably is detecting lobular and ductal equally as well. But that's something that we are going to need to explicitly experiment with into the future. And then we're also looking into sophisticating the model a bit, so that instead of just sort of normal and flag anything that's not normal, we can actually classify other things like the skin surface or if they hit muscle on the chest wall and different types of cancer within the breast. And we are seeing distinct signatures coming up for those different tissue types. So we're working on adding those into our models now.
Very interesting. So the person who asked the question says, thank you. I would definitely recommend looking into the lobular versus ductal as lobular presents so differently.
Yeah, that's a fantastic point.
Does anyone else have any questions? So for our last speaker, we have a question. Does the nanotechnology approach for glioblastoma have the potential to cure glioblastoma, or is it more to increase quality of life and lengthen the time from diagnosis to death? And can these ideas be transferred to other brain disorders?
Well, thank you for the question. It is a multi-dimensional question. My short answer would be, I hope that it's going to be a treatment by itself, but I think it has multiple possibilities. So we look at using these nanoparticles as treatment. So we would like them to intrinsically reduce the tumor burden and hopefully help to fight cancer. They can also be used as theranostics. So you can attach different drugs that are known to be able to reduce the tumor burden. And we can also use them for imaging. So to answer your question about diagnosis, these kind of nanotechnology solutions can also help to detect dormant cancer, or at the molecular level, presence of cancer even after surgical resumption. So there are many possibilities on that end as well. So I think that it constitutes a platform to explore. It's definitely there's a lot of challenge. I don't think it's gonna be a treatment for tomorrow, but I think that in combination to what we know so far, they can actually help to be complementary to the current existing treatments or imaging approaches, and ultimately they'll be beneficial no matter what for the life extent for patients.
Thank you so much. We have time for one last question for Pradarni. Does the relationship between fat and cancer apply to other cancers where Ketex is prevalent, for example, pancreatic cancer?
So my knowledge is limited in terms of the blood cancer here. I'm not too knowledgeable about pancreatic cancer or the other cancers, but I do think that adipocytes are something that we should study, because not only do they have a role in storing energy metabolism, which definitely has a role in cancer and potentially providing their fatty acids, but there is also a role with their chemokines and the adipokines, and they are involved in the chemical signaling, which some cancer cells can be reliant on. So I think it definitely, adipocytes is something that we could and should be studied in other forms of cancer, but I am not too sure about that.
Thank you so much. We're actually out of time, so we're going to be moving on to our second session. Thank you so much for all the presenters in this session.
Thank you so much. So my name is Ryan Palazzolo, and myself and Eddie Grimmett are from the Windsor Riot team, and we'll be the facilitators for session 2, which is also basic science. And we have 4 interesting talks coming up. So we'll get right into it, and I can introduce our first speaker, who is Anna Maria Lazaratos, who is an MD PhD candidate in the Department of Immunology at McGill University in Montreal. And her talk is entitled "The Impact of the Tumor Immune Landscape on Aggressive Breast Cancer." And just one other note, please feel free to drop questions in the chat throughout the talk. Anna Maria, are you ready to go?
Yes, I want to apologize in advance for my camera. I can't get it to function, so unfortunately the camera will be the way that it is. Okay, so hi everyone, my name is Anna, and my project involves studying the impact of the tumor immune landscape in the context of triple-negative breast cancer. So triple-negative breast cancer is an aggressive breast cancer subtype in which the cancer cells do not express the 3 main receptors that other breast cancer subtypes use to grow, and these include the estrogen, the progesterone, and the HER2 receptor. So given that the cancer cells in triple-negative breast cancer do not express these receptors, there are currently no available targeted therapies to treat triple-negative breast cancer. And as such, this breast cancer subtype exhibits a high propensity to metastasize, which means the spreading of cancer cells to vital organs. So the ability of cancer cells to metastasize largely depends on their interaction with various cells within the tumor microenvironment, including cells of our immune system. So this brings me to talk to you about the cancer immunoediting hypothesis, which is the first diagram at the top, which represents the various stages of the immune response in the context of cancer. So once the cancer has been initiated, there is an elimination phase during which a very important cell of our immune system called the T cell is actively eliminating cancer cells through either secreting various apoptotic factors or directly killing off the cancer cells. However, the cancer eventually employs a variety of strategies in order to escape these immune-mediated mechanisms, and these could include, for example, the overexpression of immunosuppressive molecules, one of which is glycoprotein NMB or Gpnb. So Gpnb is a molecule that is highly expressed in triple-negative breast cancer and promotes both primary tumor growth and metastasis, and also correlates with poor prognosis in patients. And our lab has shown that triple-negative breast cancer tumors in mice that do not express Gpnb, as shown in the graph at the bottom left, grew significantly less than tumors that did express GPNB. And we also found that the tumors that did not express GPNB had a significantly higher amount of T cells infiltrating the tumors, which suggests that GPNB prevents T cells from entering in order to prevent them from killing off the cancer cells. And this is important for patients because it was found that patients with advanced lung cancer that did not respond to immunotherapy had higher levels of GPNB in their blood compared to patients that did respond. And as shown by the diagram at the bottom right, we are currently assessing the efficacy of Gpnb inhibition in combination with the immunotherapeutic agent anti-PD-L1. And this has important clinical implications because if we were to administer a Gpnb inhibitor, either as a neutralizing agent or as an antibody drug conjugate that currently exists, in combination with immunotherapeutic agents such as anti-PD-L1 or anti-PD-1, in patients with triple-negative breast cancer, these patients may be able to better respond to immunotherapy. Thank you.
Thank you very much, Anna Marie, for the interesting presentation. Our next presenter will be Dr. Ken Ng. Dr. Ng is a professor at the Department of Chemistry and Biochemistry at the University of Windsor in Windsor, Ontario. And his presentation is titled Sugars and Antibodies, New Treatments for Cancer.
Okay, hi everyone. Thanks so much for the opportunity to learn about the really exciting work being discussed in this meeting. And also thanks so much for the organizers for giving me a chance to talk a little bit about our work. So as our first speaker hinted at in his excellent talk on the hallmarks of cancer, probably when we normally think about sugars, we're thinking about food and maybe things like candy and that's obviously very important for the growth of organisms and cells, especially cancer cells that need lots of energy to grow and divide. However, what I want to talk about is actually a totally different function of sugars, and this is a neglected function of sugars or complex carbohydrates. That's usually not the first thing people think of when they think of sugars. And so the picture on the left there at the top, the scary looking thing, is a cell that's covered in all types of different sugars. And in fact, all cells are covered in lots of different sugars. And in cancer cells, it's well known that there's some changes in these carbohydrates that are on the surface of cells. So our work is trying to look at specific changes in key sugars that might lead to changes in the behavior of cells, including some of the special properties of cancer cells. And one of these changes is shown in that picture just below the scary cell picture. And if you're really sharp, you might be able to see a small change. It's the addition of an acetyl group to this sugar-containing lipid that's found on the surface of many cells, especially cells in the nervous system. Anyways, this small change is found in some cancer cells, especially the cells in neuroblastoma. And this is what we've been working hard on for the past few years to develop specific antibodies that can recognize these small changes in the sugars on the surface of cells. And right now we're working with cancer biologists and clinicians to see how some of these antibodies might be able to target cells in neuroblastoma and other cancers that can lead to treatments and new ways to diagnose different types of cancer that are recognizing these very small but specific changes in the sugars on the surface of cancer cells. So thanks so much for your interest and for the chance to talk about our work today.
Thank you so much, Dr. Ng. So moving on to our next talk, we have Brandon Nault, who is a master's candidate in the Environmental and Life Sciences at Brandon University in Manitoba. And his talk is entitled, Investigating the Energy Production in Breast Cancer Cells May Lead to Identifying Disease Biomarkers. Are you ready to go, Brandon?
Absolutely. Thanks for the introduction.
Yep.
So our lab at Brandon University is trying to find out what makes aggressive breast cancers aggressive. So despite advances in both specialized treatments and early detection, breast cancer is still one of the most prevalent cancers with both a high incidence and mortality among Canadian women. So this supports the need for continued research in these areas. So what we have found are two molecules present at higher levels in more aggressive breast cancer types. Now, these two molecules have the ability to reduce the expression of multiple targets, allowing them to have a broad impact on multiple cellular functions. So to explore this impact further, we implanted copies of these molecules into the less aggressive breast cancer types to see if there are any notable changes. As it turns out, these cells display more aggressive behaviors. They grew faster. We saw increased migration and also the recruitment of the formation of new blood vessels to supply nutrients, allowing or promoting tumor growth. So in order for some of these to occur, cancer cells must be communicating with nearby cells, and they do this by secreting or letting out signaling molecules and proteins into their surrounding environment. So by analyzing what's found outside the cell, we hope to better understand changes occurring as a result of these two molecules. So what we found in this collection of signaling molecules and proteins were clues to how the cell is changing their behavior to become more aggressive, and one of which that I'm focusing on gives us a hint as to how the cell is producing energy. So this is important as the energy requirement for cancer cells is greater than that of a healthy cell, and aggressive cancers need more energy to keep up with the faster growth rates and increased migration, et cetera. So there are multiple ways in which cells can produce energy, and cancer cells are often reprogrammed to produce energy differently to account for these increased needs. So the clues that we're finding lead us to believe that the more aggressive cancer cells further alter their method of energy production to gain as much energy as they can, as quickly as they can. So the intentions of my research moving forward are to explore the specific pathways and molecules involved in these altered energy production mechanisms. And this investigation has the potential to uncover how we could safely interrupt these energy production lines to possibly slow down the cancer or limit the aggressive behaviors of these cells. Also, by exploring what these cells are secreting into the surrounding space, we're increasing our chances of identifying blood-based biomarkers. So we've already shown that the two molecules we found in aggressive breast cancers have biomarker potential and are detectable in the blood. So we're investigating downstream changes induced by these molecules in hopes of developing a battery of multiple markers to increase the effectiveness of any developed screening test in the future. Thanks everyone for your time.
Thank you very much, Braden. So our next presenter is Olivia Sell. Olivia is a PhD candidate in medical biophysics at the Schulich School of Medicine and Dentistry at Western University in London, Ontario. And Olivia's presentation is titled Imaging Tumor-Associated Macrophages with Fluorine Mri.
Hi there, thank you for the introduction, and I'd like to start by acknowledging my supervisor, Dr. Paula Foster. Most people are unaware of the fact that there are many cell types in a tumor, not just the cancer cells themselves. For example, in breast tumors, there are adipocytes, fibroblasts, endothelial cells, lymphocytes, and macrophages. So it's a very complex environment that we're still trying to understand. This talk is about tumor-associated macrophages, which I'm going to be calling TAMs. TAMs are extremely important, and they play an important role in this environment. They're responsible for helping the cancer to grow and spread to other areas of the body. From clinical analysis of human breast tumors, we've learned that the more TAMs there are in the tumor, we would expect worse patient outcomes. So our lab is developing methods to image TAMs and figure out how many of them are in breast tumors. Our research is in mice that have breast tumors. We administer an MRI contrast agent, and this agent from the bloodstream is taken up by TAMs. Then we can do a special type of MRI called Fluorine Mri, which images the specific contrast agent. In a first project, we looked at imaging 3 different types of breast tumors. One of them is a non-aggressive breast tumor, and another is a very highly aggressive breast tumor. These would all look the same anatomically, but we can see the Fluorine Mri signal, which is shown on top in magenta, that the most aggressive tumor has the most signal. And this signal is directly related to the number of TAMs that are present. So this MRI technique can be used as a diagnostic tool to determine which breast tumors are the most aggressive. Another project we're studying is breast tumor growth and development. In the MRI scans, we can see that these tumors are getting larger over time. And in the Fluorine MRI scans, we can see there's significantly more signal, indicating that there's more TAMs present at these later times. So we can also use this imaging tool to see how far the tumors have progressed over time. Currently, we're looking to image the TAM response to cancer therapy. Because TAMs are considered bad, there are new treatment options to target these cells. And in this study, we're using a drug called cladronate. We can see from these full body images that the tumors have less signal when using this treatment. And so overall, this work shows we can monitor cancer treatments with this MRI method. Overall, Fluorine Mri gives us important information about TAMs. We learn about where they are, how many there are, and we can also study the aggressiveness, growth, and treatment of cancer. Thanks for the opportunity to speak.
Thank you very much, Olivia, and thank you to all of our presenters. We can now open the floor up to any questions. The question and answer period will last until approximately 4:05 p.m. So we have about 7 minutes for that. So we invite the presenters to turn on their cameras for the Q&A session. And if anyone would like to type any questions in the chat, Ryan and I can ask the questions to the presenters. So our first question is for Olivia. And the question is, does imaging with fluorine-19 change depending on the stage of the cancer?
Yes, it certainly would. So this is something we're investigating. In this top figure, we're looking at the difference between aggressive and non-aggressive cancers, but we're also interested in studying how the aggressiveness changes over time. And so, yeah, this is definitely a focus of our work, and the Fluorine Mri method is capable of distinguishing these different types of tumors.
Great, thanks, Olivia. We have another question in the chat here for Anna Maria. So what do you see as the role of dendritic cells in the breast cancer immune landscape?
So for dendritic cells, we are not necessarily looking into those, as we are mainly macrophages. Dendritic cells are important because without dendritic cells, we are not able to activate the T cells. So they are crucial in the sense that you need them in order to be able to activate the T cells. It's not necessarily something that we see a difference between in tumors that lack versus express Gpnb. Basically, we don't see a difference in dendritic cells between the tumor types, but dendritic cells are obviously a very important component of the immune response in the sense that without them, we can't activate the T cells, and immune-mediated tumor cell elimination would be hampered a lot.
Great, thank you very much. So our next question is for Braden, and the question is, does the microRNAs do the microRNA types change with the stage of the cancer?
Thanks for the question. So we saw minimal differences in the expression of these microRNA regarding the different stages. So what we did see was more of a difference in their expression based on the cancer subtype, so based on their hormone receptor status. So there is some possibility of a subtype specific effect, which we'll be further investigating down the road.
Thank you. Now we have a question for Ken. So what are some other types of cancer immune therapy, and how does your sugar antibody method compare?
Right, so I think yeah, most immunotherapy is targeting the response of the immune system and sort of activating it towards cancer cells. So yeah, our approach is to identify a specific target that can then deliver maybe specific drugs or maybe radiation treatments to the specific cancer cells that have the modified carbohydrates that we're studying in the cancer cells. But it's definitely related to sort of a lot of the immunotherapy approaches, in that activating the immune system would then sort of activate response of like the antibodies that we have that are specific for these cancer-specific targets.
Thank you, Dr. Ng. So the next question is from Colleen, and her question is similar to the one she asked before. And her question now is, have they looked at the detection/grading for lobular breast cancer versus ductal? because lobular can be very hard to see in a lot of imaging. And this question is for Olivia.
Thank you for the question. No, we've not specifically looked at those differences, but I wanted to thank you for challenging us to do that. We continue to test new things, and so that could be a future direction for us.
Thank you. Then we have a question in the chat here from Dr. Freeman asking to Olivia as well. Given that mammography is the standard screening test for breast cancer, any application outside of MRI? Given that mammography is the standard screening test for breast cancer, is there any application outside of MRI?
Yeah, it's a great question. I would not expect that this fluorine MRI treatment, or sorry, imaging would ever replace the standard mammography. It's just not super practical. So right now we're just using it to study treatments and the development in a preclinical setting. There are other MRI agents that are iron-based that are being used in a variety of different clinical trials. One has been recently conducted in glioblastoma, actually. And so using MRI to study TAMs is being done in humans more and more recently. So it'll probably be mostly in a research setting at this point.
Thank you, Olivia. We have one more question for you, actually, Olivia. And the question is, how does MRI compare to normal MRI compared to fMRI?
Okay, yes, so fMRI stands for functional MRI, and it's quite a completely different technique, although we can use the same machinery. So fMRI is looking at hydrogen nuclei, whereas the fluorine imaging is looking at specifically detecting this contrast agent. So I don't have a lot of time to explain it, but they're completely different besides the fact that they're using the same machinery.
Thank you. And just as we wrap up session 2 here, I just want to say thank you to all of our speakers. And I'll turn it over now to Claire Park, and she will introduce our next session. Thank you.
Thank you so much. Yeah, so thank you so much, everyone, for your engagement during those 2 sessions. And special thank you to our Montreal and Windsor Riot team members for moderating those sessions. So I'll now share a Zoom poll for the Basic Science Audience Choice Awards. I will do that right now. All right, so please feel free to vote for your favorite faculty scientist talk and your favorite student talk. We will then take a short 5-minute break before starting our translational cancer research talks. And Corey from the Toronto Riot team will start the session in 5 minutes at 4:10 p.m. So please feel free to continue engaging in the Zoom chat function, and our speakers will try to answer any questions as needed. So we'll see you in 5 minutes.
All right, so with voting continuing, I'm going to introduce our next section. My name is Corey Richmond, and I am a PhD candidate at the Labatt Brain Tumor Research Center affiliated with Sick Kids Hospital, and I am representing the Toronto division of Riot, and it is my pleasure to introduce session 3, looking at clinical and translational research. And it's very fitting that I'll be presenting Dr. Artie Lutchman, an adjunct professor and scientist at the Arnie Charbonneau Cancer Institute at the University of Calgary, who will be giving a talk entitled Glioblastoma Brain Tumor Stem Cells, Biology and Experimental Therapeutics. So without further ado, the floor is yours.
Thank you, Ryan. Are you, sorry, Corey, you're loading my screen. Yeah, thanks. Hi, so I'm Artie Lutchman, and thank you to the organizers for giving me the opportunity to give this talk. So I work on glioblastoma Gbm, which most of you have heard about. It's been in the news lately, obviously. There's a lot of celebrities, people like Gord Downey in Canada, who've been affected by this disease. So glioblastoma is the most common adult primary brain tumor. And primary just means that it happens directly in the brain. It's not metastases. It's the most common brain tumor, but it's not necessarily the most prevalent common cancer. It's actually fairly rare. There's about 3 in 100,000 people that are typically diagnosed per year, but it's a devastating cancer. So it's actually the survival time is quite narrow in the sense that you can get the first tumor and then there's surgery, intervention, there's chemo, radiation therapy, but the tumor always comes back. So the median survival is only about 15 months. And also the age at which people get it. There are some pediatric, there's some adult, young adults, but typically the age of incidence for adults is between 55 to 65 years. So sort of when people are just kind of, middle age, finishing their careers. And so it's a devastating kind of time to be, to get this cancer. Tumor stabilization itself is very difficult to achieve because the disease comes back, despite all of the care that's given. So our group works on this idea that's been shown that there are specific cell types in this tumor, which we call brain tumor stem cells. And cancer stem cells are a very fitting sort of thing. Just recently, John Dick, who had discovered cancer stem cells, was given the Gardner Award yesterday. It was awarded the last couple of days. That was the discovery that there are cells in cancers that can act as stem cells. So they're not necessarily stem cells as in regular stem cells, but they're stem cells to the cancer in the sense that they can generate the disease. They also can hide from therapies. So we work on these models that where we have surgical samples that come to our lab from which we can establish these cells in culture. And they have these features where they can actually generate different cell types once you grow them. They self-renew in the sense that they can replicate themselves. And then if you put them into mouse models, they will generate tumors in the mice. And so the way the culture techniques are used are the same ones that were identified by Dr. Sam Weiss, who I work with, for neural stem cells. And they were applied by Dr. Dirks from Toronto, from Sick Kids, to be able to culture using this system. And these cells, as I mentioned, are hypothesized to underlie the tumor formation and the resistance to treatment, to current treatments. Therefore, our thesis that we explore is really that it's really important to design therapeutics that would target these cancer stem cells, these brain tumor stem cells, in order to derive therapies that are going to be effective for GBM. So we use these models in culture and in vivo, in immune deficient mice, to grow these tumors, and then we apply therapeutics to them. So we've actually worked very closely with Dr. Dirks' group as well, and we got funding from Stand Up to Cancer, so charitable organization, Terrafox Foundation, where we actually have been able to use funds to test new therapeutics or new medication in in these animal models to try and study how to target these cells. But more than that, we also study how these cells function, what's their biology, what makes them different. We do a lot of drug testing. We also do a number of assays where we kind of try to see what makes these cells what they are. And basically, we are trying to design to take some of this research or some of these findings into clinical trials. And we've successfully actually translated some of these findings through collaborations within Canada into clinical trials to see if we can test these novel therapies. Thank you.
Wonderful talk. Thank you so much, Dr. Lechman. Moving on to our second talk, I'm going to be presenting Cindy Ha, who is a PhD candidate in the Department of Medical Biophysics at the University of Toronto, and she'll be presenting a talk called Tuning the Radio, Radiation Therapy and Immunotherapy in Anti-Tumor Immunity. Go ahead.
All right, thanks, Corey, for the introduction. So hi, everyone. My name is Cindy, and I am a PhD candidate at the University of Toronto. My research looks at how our body's viral responses actually contribute towards anti-tumor immunity. And so my project actually models late-stage breast cancer, as this type of cancer tends to be more difficult to treat. In the clinic, there are limited treatment options for late-stage breast cancer. And so one of the reasons why this is so difficult is because the immune cells inside the tumor often have a tough time finding and fighting the cancer. So my research aims to improve our body's immune response to cancer by radiation therapy and immunotherapy. Now, radiation has been used to treat cancer for quite some time now, and it's been proven to be very, very effective for site-specific treatment. Immunotherapy, on the other hand, is very new, and we're still learning much about it. Our group, amongst many others, have shown that adding immunotherapy to radiation induces anti-tumor effects, not only to the site of treatment, but to tumors located across the whole body. Thus, we've been able to show that radiation and immunotherapy can induce systemic anti-tumor effects in mice. We've shown that this response is partially due to an arm of our immune system that is triggered by viruses. Both viruses and radiation therapy are able to induce the release of DNA, which can activate a viral response pathway called cGAS-STING. cGAS-STING will induce a strong inflammatory response throughout the body, leading to tumor shrinkage in our mice. Overall, these findings show that the viral cGAS-STING response pathway is necessary to achieve strong anti-tumor immunity. Recent findings in my work have shown that DNA released from radiation can actually also stimulate another kind of immunity, that being bacterial responses. And so this DNA can activate this antibacterial response inside of the cell that will then be able to send help to immune cells to strengthen the anti-tumor response. And this is by messages or care packages, as you see in this slide. And so my research so far, my early work, shows that these antibacterial responses may be an important player in mediating these anti-tumor effects. Overall, the central question of the PhD is to understand how these viral and bacterial response pathways contribute to radiation and immunotherapy-induced anti-tumor responses. By better understanding how systemic anti-tumor immunity can occur, we can work towards creating more treatment options and improving the quality of life for patients with late-stage breast cancer. Thank you for listening.
Great talk, Cindy. And moving on to our next speaker, I want to welcome Dr. Julia Bernier, who is a PhD scientist at the Gerald Bronfman Department of Oncology at McGill University, and she will be presenting a talk entitled Circulating Tumor Molecules in Liquid Biopsy.
Thanks, and first of all, thank you to the organizers for the invitation to speak. I'm going to talk a little bit about what my research program is on, and my interest is really on metastatic disease, how cancer cells leave their primary site and the disease progresses and eventually leads to metastatic disease, which we know is responsible for the majority of cancer-related deaths. And one of the challenges that we face in studying and also in the clinical surveillance of disease progression is that tumors are really quite heterogeneous. They differ in terms of between patients. Even two patients with the same tumor type can have very different molecular profiles. And even if we look at a particular patient, the cell populations in a particular mass can differ substantially, and even more so when we talk about different masses in the same patient. So that heterogeneity is then further confounded by the fact that when we treat patients and over time, the tumors evolve and change further. And that heterogeneity is very difficult to capture. What we rely on for our research is tissue biopsies, which are a snapshot of a particular tumor and of a particular disease. And so to circumvent this, my research program focuses heavily on using liquid biopsy analytes, which very simply put are the sampling of biofluids, like the blood or saliva or urine, where from those samples, we can detect tumor-derived molecules. And that can be things like circulating tumor cells, circulating nucleic acids. My lab focuses a lot on ctDNA, which is circulating tumor DNA, and extracellular vesicles, which has been mentioned already in this series, which I like to think of as small representations of the tumor cells. And we know they play very active roles in tumor progression and metastasis. And what this gives us is insight into tumor heterogeneity, because if we use, for example, the blood, it really is representative of all the different populations of cells coming from all the different potential tumors or micro tumors that are present in a particular patient. And my lab does this for two main purposes. One is to identify biomarkers, and we work heavily with circulating tumor DNA to link it to how we can use it to monitor patients on treatment, to identify mechanisms of resistance. For example, if mutations pop up over time that might not be in the primary tumor that was sampled using a biopsy, but that emerges over time and could be responsible for treatment failure in that particular patient. So we really use ctDNA really as a biomarker discovery tool to be able to monitor and for the surveillance of patients in a really personalized, precise way. And the second part of my laboratory focuses on using therapeutic targets, looking for those using particularly extracellular vesicles. And what we do here is to create synthetic vesicles and then load them, sort of recreate the ones that come from tumor cells, load them with different proteins and different DNA fragments that we think could be important, and then test them. And that allows us to identify potential targets that we can then use for therapeutics. So thanks for listening, and I'll be happy to take questions.
Wonderful talk. Thank you so much. And finally, I want to present our final speaker for this particular session, who is Emily Carpentier, who is a PhD candidate in medical biophysics at the University of British Columbia, and she'll be giving a talk on no need to hold your breath for a cure. So it's all yours, Emily.
Thank you for the introduction. So I'd like you all to take a moment and think about your breathing. As your lungs expand and contract, they're moving the organs around them. In particular, your liver, the blue organ on my slide. For liver cancer patients, this breathing motion can actually be a serious problem. Last year, over 3,000 Canadians were diagnosed with liver cancer. And on average, only 19% of them will survive 5 years after their diagnosis. About half of these patients will receive radiation therapy, where a large machine aims beams of radiation into the patient's body to kill their tumor while avoiding the organs that are nearby. So if a patient's liver is moving, how can we safely treat them with radiation? Well, thankfully, here in Vancouver, BC, we have an advanced radiation machine that can do tumor tracking. It can tilt the radiation beam to follow or track a moving tumor while the patient breathes and continuously irradiate it. But there's a problem. When we prepare for radiation treatments, we first put images of the patient's organs into a computer, shown at the top of my slide, which then predicts where the radiation would go in their body during treatment. The computer calculates if we can kill the cancer cells while keeping the organs nearby safe. And since these images are taken at one point in time, we call this a three-dimensional or a 3D prediction. And it works well when the tumor and organs are not moving during treatment. But during tumor tracking treatments, there's a lot of motion. The beam is tracking the tumor, and the patient's breathing is making their organs move. Since this prediction is based on just one snapshot in time, we won't know if at other times an organ will move too close to the beam and receive too much radiation. So to address this, I developed a new prediction method that uses 10 snapshots of the patient's organs over their breathing cycle, from inhale to exhale and back again, which is shown at the bottom of my slide. With this extra dimension of time, my 4-dimensional or 4D prediction knows where the organs move to at different times, which makes the radiation prediction more accurate. And next, I wanted to test this. I ran 3D and 4D predictions of 10 patients' radiation treatments. The 3D method showed that all of these patients' organs would be safely below their radiation limits during treatment. However, my 4D method showed that actually, at some point while these patients are breathing, about 1 in 5 organs would get too close to the beam, exceed their radiation limit, and could be damaged. Because of my research, the Vancouver Cancer Centre now uses a version of my 4D method before any patient has a tumor tracking treatment. And so now we can safely use tumor tracking technology to save the lives of liver cancer patients. Thank you for listening.
Great talk, Emily. So if all the speakers could please have their videos on, I'm going to jump to a couple of questions that have come up in the chat. And the first one is directed to Cindy. And the speaker wants to know, Randy wants to know whether a vaccination could induce enough of an immune system response to generate the benefits that you had found.
Hi, Randy. Great question. So I definitely think so. I think it would really depend. I mean, if you want to ask about whether or not it'd be C-gasting mediated, it really would depend on what is inside of the vaccine and like what is constituting the vaccine itself. If it's DNA, then it'll probably induce C-gasting, but there are many other signaling pathways that will activate when things like RNA are inside the virus. Hopefully that answers your question.
Great. And then I'm going to move on to another question from the chat from Rosaline. And this is directed to Dr. Bernier. And they want to know whether you can say if a tumor markers are low. So this is largely addressing the limit of detection and looking at liquid biopsies, whether they represent the mutations that are actively driving cancer growth. So at times, whether these are missed from the liquid biopsy itself.
So that's a great question and a complex question that requires a complex answer that I will try to be brief about. It's when we talk about tumor markers in the blood, such as CEA and CA-125, which are wonderful markers that we use clinically for patients, we know that there are a number of cases that will lead to normal numbers despite disease progression, despite substantial tumor disease. When we talk about ctDNA, it's a much more specific test, which of course requires very high sensitivity, more so than protein-based assays. There's a number of reasons why ctDNA might not be picked up. One is because, again, the tumor sampling that we do is generally based on a very small area of a tumor tissue. And these mutations that can be identified in those biopsies or resections might not be everywhere in the tumor. It also might be that what is responsible for tumor progression might not have those particular mutations that are used to pick up ctDNA.
Terrific. So just for the interest of time, I'm going to let us move on to our next session and pass it over to Komala and to Claire.
Okay, we'll get started on our final session for today then. On clinical and translational research. This session will be moderated by me. I'm Kamila Zakirova, a PhD candidate at Western University, and by Claire Zhang, who is an MSc candidate also at Western University, and we are members of the London Riot team. And I have a pleasure of introducing today Dr. Elisa Vito, who's a senior scientist in preclinical development at Point Biopharma, Toronto, Ontario. And Dr. Vito's talk is titled Radioligand Therapy, the next chapter in precision oncology medicine. Go ahead.
Thanks so much. So we often think about cancer as really being one disease, but it's important to know that cancer itself is really just an umbrella term for more than 200 diseases that all fall into this general category of having uncontrollable cell growth in a part of the body. And for this reason, it's really challenging to think that we will ever be able to have a single type of therapy that's going to be able to treat all cancers. So there's been over a century of research into the use of radioactive molecules, and this has led to the development of an innovative class of therapy called radioligand therapy. So this therapy is a form of precision oncology, and it delivers treatment directly to cancer cells in a targeted and precise manner. So I want you all to think about all of the cells in your body as being like little ships in an ocean that are constantly releasing green gas as they work. The cancer cells are also cells that originated in your body, and therefore they too would also be releasing this green gas, which often is what makes them so hard to identify as being bad by the immune system. But one thing that distinguishes through these mutations, we can imagine that those cancer cells, as those little ships, would now, instead of just releasing green gas, might also emit blue gas. And then what if we could develop a therapy to selectively target that blue gas? And this is the basis of targeted radioligand therapy. So a radioligand is composed of three parts. There's the cancer targeting molecule, which is essentially the thing that's going to target that blue gas, a small chemical linker, and then a medical radioisotope that's going to emit radiation directly to the surrounding tissue. And this radioactive missile, as we like to think of it, is injected into a patient. It seeks out the cancer cells and it delivers radiation directly to the tumor, meaning that healthy cells go largely unaffected. So at Point Biopharma, we are working on the development of many different radioligand therapies right now, one of which is currently in a phase 3 clinical trial called SPLASH for patients with metastatic castration-resistant prostate cancer, which is a form of advanced prostate cancer that has spread to other parts of the body. So again, if we imagine that all of the cells in our body are being like those ships floating in the ocean, we can now think of the prostate cancer cells as emitting these huge amounts of blue gas. And the difficult thing when trying to develop targeted therapies is always trying to identify what that blue gas is. And for prostate cancer, we know that it's something called prostate-specific membrane antigen, otherwise known as PSMA. So we target PSMA with our targeting molecule, which is linked to a radioactive isotope called lutetium-177. So our targeting molecule binds to the PSMA on prostate cancer cells, and we get this accumulation of lutetium-177 at the tumor, which subsequently delivers toxic levels of radiation to the cancer cells and kills them. So radioligand therapy and precision oncology is really rapidly becoming an integral part of cancer care, and the FDA just recently approved a radioligand therapy for prostate cancer patients. Radioligand therapy can significantly improve a patient's quality of life, and at Point, we are actively working to engage patient partners in our strategies and to educate the public about these new and exciting therapies. Thank you.
Wonderful talk, Dr. Vito. So moving on to our next presenter, it's William Make, a PhD candidate at Biochemistry and Biomedical Sciences at McMaster University, and Make's talk is titled Targeting the Core of Glioblastoma.
Thank you for the introduction, Camilla. So I'm a PhD candidate in the lab of Dr. Sheila Singh, and here we study multiple types of brain tumors, or brain cancers rather. And specifically, my talk today will be about glioblastoma, which has been extensively discussed today, so I won't go too in depth on the background of it, but as has been mentioned, it's characterized by being extremely aggressive, extremely resistant to typical therapeutic strategies such as surgical resection, chemotherapy, and radiotherapy. And the 5-year survival rate is about 6.8%. So it's quite a devastating disease. So the ability for Gbm to become so treatment resistant and to exhibit this extensive tumor heterogeneity is thought to be caused by, in part, due to cancer stem cells or brain tumor initiating cells. And in figure B here, we see that in 2004, my supervisor, Sheila Singh, who at the time was doing her PhD in the lab of Dr. Peter Dirks in Toronto, she found that when she cultured these patient-derived GBM cells, the ones that express CD133 were capable of growing quite aggressively and forming tumors both in the cell culture dish as well as in vivo animal models. But the ones that didn't express this CD133 protein were not capable of doing so. So this was kind of the first putative marker of these brain tumor initiating cells or cancer stem-like cells that initiate glioblastoma. And so our lab's role, in recent years, there's been more studies that have come out showing, well, CD133 is not the be-all end-all of brain tumor initiating cell markers. And there are CD133 negative cell populations from different GBMs that are capable of possessing these brain tumor initiating cell-like traits. So in our lab, we're aiming to identify more of those markers, and we do this by collecting patient tumor specimens. And as has been previously described, we process these specimens and then turn them into a cell culture line, after which we're able to perform a variety of different experimental analyses on these to discover and elucidate different putative brain tumor initiating cell markers, one of which we recently found to be a cell surface membrane marker called CD70. And another aspect of what we do in our lab is we develop a therapy called chimeric antigen receptor T cells. And this is done by acquiring patient blood and isolating an immune cell called T cells, at which point we're able to re-engineer these patient's immune cells into these CAR T cells, which specifically target an antigen of interest, such as CD133 or CD70. And when we culture these cells with GBM cells that express CD133 or CD70, we see significant killing of those cells in the cell culture dish. But in figure E here, we also see that when we intracranially inject mice with these patient-derived GBM cells and then treat them with our CAR T therapies, we have a control CAR T that doesn't target any particular cell surface membrane that's on GBM. And we have our CD70 specific or CD133 specific CAR Ts that can target either of those two tumor antigens. We see a significant reduction in brain tumor volume, as can be seen here in figure E. We see on the left, the mice treated with the control CAR T exhibit quite a large brain tumor, whereas the CD70 CAR T treated mice have essentially complete eradication of the GBM tumor. And as can be seen in figure F, and as would be expected, the mice who are treated with the CD70 CAR T fare far better than those treated with the control CAR T. So we're still looking at for a variety of more of these brain tumor initiating cell markers, because we believe that there's not going to be a one and done therapy for GBM, unfortunately. And so we're now aiming to combine these therapeutic strategies into a polytherapeutic strategy to really target the core of GBM, which is these brain tumor initiating cells, which end up seeding recurrence and evading therapy. And we're now working closely with a couple of biotechnology companies in order to bring these therapies to patients as soon as possible. So thank you for listening to my talk, and I'll be glad to take any questions at the end of the session.
Wonderful talk, William. So moving on to our next presenter, Dr. Louis Hua Tai, who's Associate Professor in Cancer Immunology at University of Sherbrooke, Director of Cancer Research Access, and Dr. Tai's talk is titled Cancer Killing Viruses to Treat Bladder Cancer. Please go ahead.
Thank you very much, Camila, and thank you for the invitation to talk about my research this afternoon. So it's my privilege today to talk about cancer killing viruses I know it's a difficult time to imagine how a virus can be used for a good cause, especially since we're still in a pandemic, struggling with the SARS coronavirus that has caused considerable harm on a global scale for everyone. However, I would like to impress upon you today that not all viruses are bad. Most of them are benign, and some of them can actually be beneficial. And in fact, millions of viruses coexist with us, and they cause us no harm. They live in our gut, on our skin, in the air that we breathe, in the water that we drink. They're everywhere, and they're even integrated in our human genome. They're called endogenous retroviruses. So anyway, we can actually engineer viruses in the lab for beneficial purposes. We can turn them into mini biotherapies to target cancer. And so how do they work exactly? So we can build viruses to specifically target cancer cells while leaving normal cells intact, unharmed. And in doing so, they can actually then activate the patient's own immune system, which obviously in the context of cancer, or specifically in bladder cancer, it's not working properly, right? Because your immune system can no longer see the cancer, and your cancer has proliferated and has spread. So in fact, these viruses or biotherapy is a form of immune therapy, because we're using this entity to activate the patient's own immune system to fight back against its own cancer cells. And this is the basis of all immune therapies. And in fact, in bladder cancer, a very old form of immune therapy called BCG therapy has been used for the past 60 years. BCG stands for Bacillus Calumet-Guérin, and it's a lab-engineered bacteria that has been used to treat bladder cancer that has not spread into the muscles surrounding the bladder, so for non-muscle invasive bladder cancer. However, we found in our lab studies that our viruses, our beneficial viruses that we've engineered, can work better than these BCG bacteria, which is standard of care, and that they're better able to target bladder cancer cells, whereas BCG indiscriminately enters into both healthy cells and bladder cancer cells. So in the far left panel, we are developing a form of beneficial viral therapy that can be placed directly into the urinary bladder via the urethra, where it can do two things. It can directly kill bladder cancer cells, so we're moving into the middle panel, and two, it can also activate the patient's immune system that are attracted by the proteins that are released by the bladder cancer cells as they are being infected by the virus. And then moving on to the last panel on the far right, these immune cells will communicate with each other in an intimate way, and then go back to target any remaining bladder cancer cells in the bladder, or potentially even metastatic cancer cells that have escaped beyond the muscle walls of the bladder. And it's hoped that this new virus-based immune therapy can be used to treat bladder cancer patients that have failed their frontline surgery, that have failed their BCG therapy, or perhaps even to replace BCG entirely. Thank you for your time.
Thank you so much, Dr. Tai. It's a wonderful presentation. So moving on to our last, but definitely not least presenter, Tricia Chenery, who is a PhD candidate in biomedical biophysics at Western University, London, Ontario. And her talk is titled, "Images are more than just pictures, they are data."
Thanks for the introduction. So by now, we're all familiar with how important medical imaging is for the visualization of structures within the body. We know that it is commonly used in clinical practice to diagnose and treat cancer. But what if I told you there was a way to utilize these standard medical images to extract numbers? This numerical data could not only help provide a more comprehensive view of cancer, but predict outcomes for patients as well. The field I'm describing is known as radiomics, and it aims to develop, evaluate, and translate clinically relevant image analysis tools to support diagnoses and treatment. Generally, the process involves acquiring medical images through CT, which can be thought of as a 3D X-ray, and then identifying the regions of interest. So in our case, these would include our cancerous tumors. Next, we would extract radiomic features such as shape, size, and texture that describe these regions of interest. We'd collect large data sets, and finally, we'd use machine learning models to discover patterns within these large data sets. My lab is currently looking into using radiomics on the CTs of oropharyngeal cancer patients. Oropharyngeal cancer originates in the back of the throat, and it's the most common head and neck cancer. It's currently treated with radiation therapy, and the CTs we study are the images taken prior to receiving radiation in the clinic. The good news is most oropharyngeal cancer patients have a survival rate of over 80% after treatment. The bad news is almost all patients will suffer treatment-related toxicities such as dry mouth and trouble swallowing. Additionally, approximately 30% of these patients will require a feeding tube to combat weight loss. Physicians want to avoid giving all patients feeding tubes as the insertion is an uncomfortable, invasive procedure. So it's important to know who will require a feeding tube the most. Currently, physicians have a hard time determining which patients will require a feeding tube, and therefore we have this unmet clinical need to identify the high-risk patients when they first get scanned in the clinic. Accurate radiomic features could assist in identifying which patients could benefit from a feeding tube prior to the start of their treatment. Using our extracted radiomic features, we can teach a computer to learn the difference between number patterns in high and low-risk patients. Once the computer is trained, our goal is then to build a machine learning model to aid physicians in clinical decision-making. This non-invasive tool would be the first of its kind to integrate information from medical images, patients, and their treatments, leading to more personalized supportive care options and ultimately improving quality of life for cancer patients. Thanks for listening.
Thank you so much for all the presenters. We're moving on to the questions period, and please feel free to ask your questions in the chat.
Okay, so we do have a question for Dr. Alyssa Vito, and the question is, compared to other cancer treatments, what kind of patients would be eligible for radioligand therapy?
So currently the only approved radioligand therapy is for prostate cancer, and there is some for thyroid cancer as well. But that being said, there is many clinical trials for breast cancer, different types of GI cancer. There's people going after brain cancer as well. So really it's a pretty broad spectrum. I would say that if anything, we get a little bit innovative about our delivery of the radioisotopes sometimes. So for instance, there are clinical trials for GBM where they actually insert a catheter into the brain directly into the tumor to ensure that there's no off-target radiotoxicities. But really, it's not a type of therapy that's specific to one type of cancer. I just presented prostate today because that's the most late-stage trial that we currently have going at our company.
All right, great. So the next question is for Trisha. And the question is, what are other cancer applications for radiomics?
Yeah, that's a great question. So radiomics can be applied to any image. So most cancer patients, when they come into the clinic, they need to get scanned on a CT so that doctors know where to treat them. So you can actually apply radiomics to any medical image and predict outcomes for cancers of all types.
Thank you. So the next question is for Dr. Li Huatai, and the question is, can you elaborate on the translational models of bladder cancer?
Sure. So in the lab right now, we have mouse models where they have an intact immune system, right? Because we're proposing to use a viral immune therapy. So we want to make sure that there's an immune system there to react to the immune therapy that we're putting in. So that's a mouse preclinical model. There's other translational models that we're using in the lab, which involve getting bladder cancer tissue from patients, and then we expand them ex vivo into organoids. I'm sorry for the noise in the background. My son came home from school. We expand them as bladder cancer organoids, so mini bladders from patients. And at the same time, we have the patient's immune cells that we collected through their blood, and we isolate immune cells from the patient's whole blood. And we have these matched systems where we put the patient's immune system and their bladder cancer organoids that we've grown from their tissue together in a dish. And then we treat with viral therapy and other therapies to see the efficacy. And then we think that this will give us some idea if the patient's patient's bladder cancer will be susceptible to the different viruses that we're developing.
All right, thank you so much. So thank you to all the presenters and their wonderful talks, and thanks for the questions as well. I'm going to right now open up the poll for Audience Choice Awards, and I will be passing it on to Claire.
Awesome. So thank you again to all our incredible translational cancer research speakers and for your engagement during those 2 sessions, and also the Toronto and London teams for moderating those sessions. So as Claire mentioned, we have now launched the final Audience Choice Awards poll for these 2 sessions focused on translational cancer research. So please select your favorite scientist and professor and student talk, and I'll just give you a quick minute to do so. It's already 30 responses. So in the meantime, that brings us to conclusion to our first National Rights Symposium, Choosing Hope Canadian Cancer Research. And I'd really like to reiterate and thank all our speakers. A special thank you goes up to our keynote speaker, Dr. Fred Dick, for sharing about the hallmarks of cancer. For all our 3-minute speakers, delivering these short and engaging talks with the aim to make it very accessible and understandable to the community is a really big challenge that we think that you all executed really well. And that was very clear from the quality of the talks this afternoon. So we would really like to commend our speakers for their truly amazing talks. And that being said, I would like to pass on the floor to my co-chair, Olha, to share the Audience Choice Awards for the best speakers for both the basic science and translational research sessions. We might give her a minute to do so since she's updating those slides. And I do encourage you, if you do have any further questions, to just reach out in the chat as well. If our speakers aren't here to answer them, we can redirect you to contact them as well.
Okay, thank you, Claire. I am just pulling up the presentation. Okay. For our first Audience Choice Award, thank you so much, Jessica, for being our favorite basic science professor, clinician, and scientist talk. Jessica's also a former London Riot member, so huge shout out to her.
Thank you.
For our next Basic Science Award, I'd like to present it to Ana Maria Lazaratos, who is our favorite graduate student and trainee in this session. Thank you so much, Ana Maria. For our next award in clinical and translational research for a professor, clinician, or scientist, I'd like to present this award to Dr. Liwa Tai. Thank you so much for your very informative talk. And lastly, for our Audience Choice Award for Clinical and Translational Research for a graduate student and trainee, I'd like to present this award to Trisha Shinry, who's also a London Riot member.
Thanks, Ola.
Perfect. So at the very end now, I'd like to thank all our speakers for all your fascinating and incredibly informative talks. I'd also like to thank everyone on the Riot team for helping organize this event. In particular, Claire Park, Claire Zhang, Camilla, Corey, Eddie, John, Morgan, Patty, Agatha, and Sandrine. I'm really hoping I didn't miss anyone now. But thank you all for all your help in putting together this great event. And as well, thank you to the Canadian Cancer Society for all their support of Riot and this symposium. Now, lastly, I'd like to thank all of you so much for listening and tuning in with us this afternoon. I hope that the symposium highlighted how leading scientists and trainees are working hard to address the complex challenges in cancer research. I'd like to remind you to always seek reliable cancer information. So the cancer.ca website and the 1-800 number listed on the slide are really great sources to start from if you have questions. If you enjoyed the symposium, we also encourage you to check out the local Riot team's websites to learn more about their other similar initiatives and to follow them on social media as well. We will be posting the recordings of the symposium today on our website. So if you did enjoy your time today and want to share the talks with anyone else, feel free to do that. Thank you everyone once again for joining us and enjoy the rest of your weekend.